Hello, peeps. The camera on my computer is horrible, so I'm trying to get a lighting situation where you can actually see my face. I usually look like a ghost. Right now, it's pitch black in this room, and the only light is coming from the screen. Seems to be the best. All right, so I wrote notes for you for this new chapter. I did not load them up on Moodle yet, so I'm going to do that right now as we speak, as I speak. You guys aren't speaking. Okay. Doing it. Turn editing on. We are Santa cells, filling Santa's shelves. We are Santa's elves. I have no idea where that came from. Okay.
It's from the Rudolph movie.
I know, but I don't know how it popped into my head just now, but there it is.
Probably from the snow.
It's from Rudolph the Red-Nosed Reindeer, Rankin Bass in 1967, I believe. So I just uploaded the notes. They're the last thing in the block. Let's put it up where it belongs. Dawn, lift us up where we belong. That's a bad joke. Okay, coming up. Chapter 23. Oops. Actually, I have some new slides that aren't included in the PowerPoint that's up there, but I'll put that up later. So we can go ahead and get started today. What kind of attendance we got today? See how we're doing. How are we doing? Okay, 39. That's not bad. I think there's about 100 people in the class. No, I think there's like 48 people in the class. We're doing okay. Okay, guys, are we ready to rock and roll? I have not graded your exams yet. I've been having computer problems, and I can probably come up with some other excuses if I wanted to, but basically I'm being lazy. I will
You're not in any rush.
Good to hear it. Okay, so I'm hoping to get started on that this afternoon. And today we're going to start talking about transport across membranes. And this is going to be probably going to be a short lecture. I hope you don't mind. Again, it's like if I'm in class, it takes a lot longer, and I don't want to necessarily push faster than I have in previous years. So we will do what we do today. We're rock and roll.
What chapter is this one?
Chapter 8, Transport Across Membranes. There it is. Okay, so the first picture is not important. There's nothing there that's important. I just think it's a cool picture. I think this is like, it looks like a blood clot or something. Like you've got all these fibrous particles that are trapping red blood cells. Looks like a scab. I don't know if that's true, but that's what it looks like to me. It's like a Rorschach test. That's what it looks like to me. Okay, transport across membranes. It's very important in cells. You know, the whole idea of a cell is it's surrounded by a plasma membrane, and that separates two compartments, the outside compartment and the inside compartment. So membranes are real, and then inside eukaryotic cells, you've got many other membrane compartments. And so controlling what's on either side of the membrane is really important in eukaryotic cells. And so membranes are going to block most things from moving, but you need to get things across as well. So that's what this chapter is all about. How do you get things across a membrane? Let me pull up my notes real quick to make sure I'm not forgetting something. And the key thing to remember in this chapter, okay, focus on this all the way through the chapter, is the energy. Is it costing energy to move this thing, or is it going downhill in energy to move this thing? Because we've got two big different types of transport. One is active transport that costs energy, and the other is passive transport that releases energy. And energy, if you've had biochemistry, energy is Gibbs free energy. Actually, freshman chemistry, they teach that now too. Gibbs free energy, which is both heat energy, which is what we see with ATP, a lot of enthalpy that's being released when that gets hydrolyzed, but also entropy. So a concentration gradient, when you've got a much higher concentration or just a higher concentration of something on one side than the other, that's order in the system, and that's unfavorable energetically, right? So a concentration gradient represents stored energy. And typically in the cell, if there's a concentration gradient of something, it's because the cell used energy to make that concentration gradient. Back when we were talking about skeletal muscle, we talked about the sarcoplasmic reticulum storing calcium. Cost energy to put that inside the sarcoplasmic reticulum. And then you had a really strong concentration gradient between inside the SR and in the cytoplasm. And then that was used to initiate contraction. So keep an eye on the energy in this chapter. And the first thing we're going to talk about is simple diffusion. Simple diffusion is let me pull my notes up so I don't say something stupid. Spent the whole morning organizing this, so I might as well read my notes. So simple diffusion then is just when a molecule diffuses through the membrane. It basically diffuses into the membrane and then goes through the membrane. And it can go in either direction. It's all totally passive. And diffusion through the membrane is this, basically. Some things cannot go through the membrane. Things that are polar will not go through the membrane. Things that aren't polar will go through the membrane. So I've got a little animation here that I wrote for that I made for my other class. File not found. Oh, that's right. That's gone now. Sorry, yeah, we can't see that anymore. Okay, but anyway, if something can diffuse, it has to be able to go into this lipid environment in the center of the membrane before it can go through. And for that reason, only non-polar things can get in. But to be a little more direct about it, the equation that describes diffusion through a membrane is called Fick's equation. So you can see that here in my notes. So the rate of movement across the membrane is equal to P times A times C outside minus C inside. So here's your concentration gradient, outside versus inside. And notice if it's higher inside than outside, this turns into a negative, so the rate goes negative, and basically the thing is going in the opposite direction. So concentration gradient then determines how fast it's going to move and in which direction it's going to move. P then is let's go A first because it's the easier one. A is just the area of the membrane. The more membrane, the more transport, obviously. And in terms of cells, the most important thing is your surface area to volume ratio. The more surface area to volume you have, the more transport you can go into the cell. And that's why, that's kind of the strategy of bacteria and why they're so successful, because they've got a very small surface area to volume ratio, which means they can bring a lot of nutrients in, and they can grow fast and then divide, and they can, divide every 15 minutes under optimal conditions. Whereas eukaryotic cells take, between 10 and 24 hours to divide under optimal conditions. So surface area to volume ratio is really important too for diffusion into the cell. And then P is the one that's most important to me anyway. That's the permeability factor. So that's how easy is it for that particle to get into the membrane. You know, and that comes down to two different things. One is size. So something really polar like water is extremely polar, can get through a membrane easily because it's so small it zips right through. But other things that are small are like oxygen, carbon dioxide. They all diffuse in and out of cells really easily. Those two examples are non-polar as well, so that helps as well. But size is really important. The limit of size of something that can just slip through the membrane is about 180 grams per mole, which is the size of glucose. Glucose is very polar, but it can diffuse into the cell, just not very well. I don't think eukaryotic cells could survive just on what will diffuse through passively, but it can diffuse. That's kind of the limit of how big a thing can be and still diffuse in. Now, if you look at things like proteins, there's no way a protein's going to go through a membrane. So we'll talk in the next chapter about if you've got a protein that you want to secrete from the cell, release it outside the cell, you can't make it in the cytoplasm. Once the protein's in the cytoplasm, it'll always be in the cytoplasm. So when we start making secretable proteins, you'll see you have to make them inside the rough ER, and then they're inside a membrane system. So we'll talk about that in the next chapter. Okay, and then the other thing is polarity. And polarity is basically, is it more soluble in water or lipid? Lipid-soluble things will diffuse right into the cell, no matter how big they are. Like steroid hormones are huge. Molecular weight, I'm going to make a guess, but about 350 or so. And because they're non-polar, they'll diffuse into the membrane and then through the membrane go right into the cell. And that's important for their function because they are released from specific glands, whether it's the testes or the ovaries, and gets transported all over the body, and it diffuses into the cell, and its receptor is already in the nucleus, so it has to diffuse all the way there to get there. So non-polar things will diffuse through the membrane really easily. Polar things very poorly. A lot of polar things do need to get in, and we'll talk about that in the next section. But the way you measure this polarity, whether something will diffuse or not, is by the partition coefficient. Let me change my share. Didn't mean to do that. It's okay. Here we are. So here's another little animation I made. So the idea of a partition coefficient is, if you put two different chemicals, if you put a chemical into this chamber here, you've got oil and water. Usually oil is octanol, which is like eight carbons straight with an OH on the other end. That's your oil. So if you look at these two chemicals, one's positively charged, so that's polar, and the other's not charged, and it's non-polar. You dump them in, and you can see the Y's are pretty much staying up here, but every now and then one will come to the bottom. There he is. X's too, they're mostly staying down here, but one may go up occasionally. So the idea is, what's solubility in oil versus divided by the solubility in water, and that's your partition coefficient. And that will determine, let me go back. So that is your P, permeability factor, right? Where is it more soluble? Okay, so Fick's equation then is linear. So you could, a linear equation is y equals mx plus b, right? So the plus b is your y-intercept. You could just say plus 0, because if there's no concentration gradient, there's no transport. So your rate then is y equals x. This is m. Your slope is your permeability times your area, and it always goes through the 0, the origin. So what I'm trying to say here is the rate then you put on your y-axis, concentration gradient on the x-axis, and the slope of that line will tell you what your permeability is. And you can, and by the way, just so you know, I'm not going to give you numbers so you do math with this equation. I just want you to look at the equation and see what factors are in it and understand molecularly why those factors are there. Okay, so slide 6. Watch me be multimedia today. Wow. Is that okay? Slide 6. Simple diffusion. Okay, so it's linear. The higher your concentration gradient, the higher the rate, and it's perfectly linear. Okay, that's all there is to passive diffusion. Pretty simple. More complicated, more interesting is facilitated diffusion. So like I said, there are some things that, despite the fact that they're polar, you still need to get them in the cell. Things like amino acids and sugars, right? You need to get them into the cell for energy and for building proteins. Also, there are polar things you need to get out of the cell. Cells break down proteins and they end up with urea in them, and they have to get rid of the urea. And urea is like, basically, if you took carbonic acid and you put 2 amine groups on it, very polar, small molecule. So you need to be able to get these things, out of the cell. We've got 2 different types of facilitated diffusion. One is kind of on the slow side, and the other one is really, really fast. So the slower one are transporters, and these are transmembrane proteins, so they're exposed to the inside and the outside of the cell. And they have 2 conformations. So in one conformation, they open to the outside and some specific particle will bind to it, and then they'll flip into the other conformation and release it on the other side of the membrane. And you can just imagine these things just going back and forth, and whenever a molecule happens to land in it, it goes the other way. If it landed from the inside, it will go outside, okay? So it's passive because it can go in either direction. Let's see that. Okay, so here's your glucose transporter, GLUT1. So here it is sitting in the membrane, and it's got 2 different conformations, T1 and T2. In T2, it's open on the outside, and glucose from the outside can bind. And in T2, it's open to the inside, and glucose can come out. So that's all it is. And it's not like it's just going to, transport anything in. When the glucose comes into this transporter, it binds to specific amino acids that are sticking out here that are like looking for glucose to grab it. So it's not like anything can come through. Only glucose can come through this thing. Very specific. So this then is going to be totally, the direction of transport here is totally dependent on the concentration of glucose. If it's high outside, it'll be transported in. If it's high inside, it'll be transported out. And that's really important because the cell will regulate that. We'll talk about situations when you do this and that. Another thing about this that they don't talk about in the book that's pretty cool is when glucose comes in, it's pretty much immediately phosphorylated to become glucose 1-phosphate. Maybe glucose 6-phosphate. I don't know my biochemistry as well as I used to, but basically glucose is going to get phosphorylated. Once it's phosphorylated, it can no longer bind to the inside and be passed out, okay? So that enzyme, glucose kinase, turns this mechanism into more of a one-way mechanism so that whenever glucose comes in, it gets trapped and can't leave again. And it's called hexose kinase. You might want to write that down, hexose kinase. I should put it in my notes and it'll update that. Okay. You're getting a look behind the screen, I am the wizard, the grand and great Wizard of Oz, and you're going to see how it actually gets done. Can go either way. Oh, I can't see my keyboard. Okay. Oh, I'm going to do star hexose kinase glucose transport one directional. Okay. I'll fix that later, but I want to put that in there. It's kind of like it's an interesting tidbit that you learn only because you had me for cell biology. No one else would teach you that. Considered an added benefit. Okay. Okay, so what's neat about these transporters is that it's saturable, right? You've got a molecule sitting in the membrane and it's doing this, right? You can go faster and faster, but you're going to get to a point where it can't go any faster, right? Every time it opens, it grabs a glucose, and then it closes, releases the glucose. It's going as fast as it can. Two things. That's one. I should have said this before, though. Your transport's not linear because it's being helped across the membrane. Let's do this. Let's try and talk like a human being here. Come on, baby. There it is. Okay, what I'm trying to say is your transport is not linear, okay? As you increase the concentration gradient, the rate increases in a non-linear way, much faster than it does with simple diffusion. And that's because the higher your concentration gradient, the more of these transporters you activate, okay? If you've got 100, well, let's say you've got 1,000 glucose transporters in a cell. At this concentration gradient down here, maybe only 20 of them are actually transporting because most of them don't see any glucose. It doesn't hit them properly. When you increase the external glucose concentration, now you're using more transporters. And so your rate goes up quite a bit. So it's not just linear, it's much more than linear. But then you get to a point out here where you're activating all 1,000 glucose transporters and they're all moving as fast as they can. When you increase the concentration of glucose even further, they can't go any faster. So this increase in rate here, as it's increasing, notice that it's the same slope as simple diffusion. So out here, any increase in rate is not because the transporters are going faster. It's because glucose is going around the transporters and directly through the membrane. So to understand this green line, basically, it's not just facilitated. It's both facilitated and simple at the same time. So imagine this experiment. This may make it more clear. In one experiment, you've got red blood cells, okay? You take those red blood cells and you treat them with detergent so they kind of swell up and have holes in their membrane, and you let all the hemoglobin come out, okay? And then you shrink them back down. That's called a red blood cell ghost, okay? So you take these red blood cell ghosts that are just membrane, basically, with cytoskeleton holding it together. Now, in one experiment, you put radioactive glucose on the outside, and at different time points, you take your red blood cells out, and you find out how much glucose went in, and you determine your rate of transport. So that's going to give you this line. Okay, as you increase the glucose outside, how much faster does it go inside? Okay, that's this whole line. Now you take your red blood cell ghosts again, and you block that glucose transporter with something. Maybe there's an ion that blocks it or something. Now, as you increase glucose outside, the increase in rate is just because of simple diffusion. There's no way to block that. So you've got glucose going in through the membrane directly. So this is just simple diffusion, glucose going through the membrane. This is both going through the membrane, which always happens, you can't stop it, plus those transporters working. So if you want to know what the transporters are actually doing, you do both experiments, and then you mathematically subtract this from that. And you get this, okay? So with this line, you can see right where those transporters saturate, okay? I don't know if I put this line in the right place. Let's move this over a little bit. Looks like right about there, it's saturated. So this is total transport with both simple diffusion and your transporters. This is just simple diffusion, and now you subtract this from that, and you end up seeing just what those transporters are doing. And out here, as you increase glucose concentration, you're not increasing rate at all because those transporters are saturated. Okay, does that make sense? Anyone want to chat me up on that one? If you've had biochemistry, you would have heard of this called Michaelis-Menten kinetics. Where is it? Where is it? Here it is. So it's going to saturate. It's going to be, the increase in rate is nonlinear and it saturates, and that's Michaelis-Menten kinetics. Okay. I'm not going to ask you about that on a test, but just to link this class with the other class, if you've had it before, or if you're going to have it, you'll see that come up, and that's what we're talking about. Okay, any increase in transport is just simple. So this is what I talked about. Okay, in terms of facilitated diffusion, we've got 3 different types of it. Well, I mean, I shouldn't say that. I should say in terms of transporters, we've got 3 different types. You've got your uniport, your symport, and your antiport. So uniport, it's pretty simple. It's just one thing moving. Could go in either direction if it's passive, okay? That's your uniport. Symport is when you've got 2 things moving, both in the same direction, sim being the same. Antiport, opposite. You've got one thing moving this way and one thing moving that way. So these 2 things are called coupled transport because you have to have both those things in order for it to happen. Now, what gets complicated here is symport and antiport, I believe, can both be facilitated diffusion, but sometimes they're a form of active diffusion that we'll talk about in the next class. And so let's see if we go back to your notes real quick. Okay, so uniport is one thing moving like a glucose transporter. Symport is 2 particles moving in the same direction. Now, I only know examples where it's actually active transport, and we'll talk about them in the next class. So I can't give you examples of that. I'm trying to dig some up, but I can't find them. Now, antiport could be either facilitated or active transport. You've got 2 particles moving in the same direction. We'll talk about these 2 transporters a lot at the end of the class, and these are both facilitated diffusion. So it's a passive transport. Here you've got chloride going in for bicarbonate going out, and here you've got sodium going in for potassium going out, or it can reverse and go in the other direction as well. And I think the whole idea with this antiport is charges at the membrane are really, really important. Especially with your nerve cells and muscle cells, you've got a membrane potential. So the inside of the membrane is negative. If you had to get bicarbonate in, it'd be really hard because that bicarbonate is trying to go against a negative potential. It'd get bounced out. So a transporter like this, every time the negative charge goes in, a negative charge goes out, it doesn't change the membrane potential at all. Same with sodium and hydrogen there. You've got a positive in, a positive out, and it's not going to be affected by the membrane potential. That's a little complicated, but that's basically why they're antiporting like that, because they're charged particles and they don't want to be affected by the membrane potential. Want to, like ions want or need. We know. Well, who knows? It could be small humans too. Okay, so that's one type of passive facilitated transport, which is the transporter. And again, the transporters are going to change conformation for every cycle, right? They have to keep changing transformations like that. The other type of passive transport, facilitated transport, are channels in the membrane. Talked a little bit about channels in the last chapter with skeletal and cardiac muscle. These are basically regulated pores in the membrane for very rapid movement. So what makes these so much faster is they only have one conformational change. They open And once they open, things come flooding in or flooding out, depending on, the concentration gradient of the thing that's moving. So channels are also facilitated, but they're much, much faster. And we've got a number of different types of channels here. One are ion channels. These are selective and gated, which selective means they only let one ion in. So when we were talking about the voltage-dependent calcium channel back in cardiac muscle, sodium can't go through that. Only calcium can go through it. Potassium can't go through it. So two other cations that are right there bouncing into that calcium channel will not be allowed to go through it. No negative ions can go through the voltage-dependent calcium channel. The only thing I know of that can go through the calcium channel is barium, which on the periodic chart is just below calcium. So that will also go through it, but there's relatively no barium in your body. So that's more of an experimental thing. And the other thing is the gating, which I forgot to add some things here. There are a lot of different ways these things are gated. And the gating idea is it's not always open. It's not just a hole in the membrane that allows that stuff to go through. It's opened when the cell needs it to be open for some reason. So some of the ways things are gated are by voltage. Voltage gated ion channels. We saw that in the last chapter. We also saw hormone gated, ligand gated. Okay, ligand gated channels. What else? Ligand gated, the example we gave last time was a nicotinic receptor that is open when acetylcholine's around, right? The neuron releases acetylcholine, binds the nicotinic receptor, the receptor opens. That's ligand gated. Also, let's talk about stretch activated. And here's how you can activate a stretch activated receptor. Watch this. You've got these things, these sensory neurons in your skin that are called corpuscles, Puccinian corpuscles. So they've got stretch activated ion channels in there. And when you touch your skin, the corpuscle gets pushed and stretches a little bit, and that allows ions in, starts the action potential that goes back up your spine to your brain. So stretch activator is another way to activate things. Also, cyclic nucleotide. Okay, you're doing that right now when you're looking at your screen. Your retina uses sodium channels that are activated by cyclic nucleotides. So, and I can go on and on. There's a lot of different ways they're gated, but basically they're not just open all the time. They open based on stimuli, and for that reason they're used a lot in neurons. Okay. So in terms of, this talks more about, well, I don't know, let's pull up slide 13 and see what the hell I was talking about there. Sometimes I even learn when I teach this stuff. Okay, so here's your ion channel, and this shows you why it's so fast. This is closed and that's open. So once it's open, a lot of ions can go through. Okay, so here is, I believe this is a potassium channel. Yeah, potassium channel. So here's the structure of it. Part of the structure you can see, it's mostly alpha helix, but there's one section of beta sheet that comes down the middle like this. And you've got 4 different beta sheets that come together. And so right here, you've got amino acids from 4 different beta sheets coming together like this. And the only way an ion can get through is if it interacts with those amino acids. So only potassium in this particular channel can go through. It's actually going through in this direction, going out. So that's your selectivity filter. That's why each channel is specific for one thing. I have a question in the chat. So when they are open, do they just stay open for a certain amount of time, or do they keep opening and closing like other channels? They keep opening and closing. They kind of get stimulated to open, and depending on the channel, usually they'll like open, close, open, close, open, close, open, close, and then they'll go back to their closed state. But that's typically the way channels work. Okay, so this is your selectivity, and then you've got some kind of a gate, which is down here. And with these voltage-dependent channels, basically this alpha helix here has got negative charges along it. Typically, a transmembrane region is only going to have neutral amino acids in it. But in a voltage-gated channel, you've got negative charges. So then when at rest, the inside of the membrane is negative, so that pulls this thing down and closes the gate. When you get depolarization of the membrane, like when that action potential comes down the T-tubule and it comes to that voltage-dependent calcium channel, suddenly the membrane potential changes, so it's negative outside and positive inside, and this thing comes back, it gets pulled back by that change in voltage, and that opens the gate, and then ions can go through. So this is your voltage-dependent ion channel. It's got your gate, which is voltage dependent, and then your selectivity filter. Selectivity. This is actually what the potassium channel looks like, more like this. But I had a really cool animation that showed how this works, but it's Flash. I think I told you this before. I have tons of animations that work in Flash, but they don't work on browsers today. So it's not your problem. Okay. Well, let's go back to work. We are Santa's elves, filling Santa's shelves. We are Santa's elves. Okay, so other types of channels. There are water channels. Now, I said water passes through the membrane passively, right? It can go through the membrane all by itself. It'll go through faster, though, if you've got a hole in the membrane that allows it to go through. So you've got these proteins called aquaporins, found mainly in the kidney in us multicellular organisms. Let's bring that sucker up. Oops, hang on. Here's your aquaporin, a really big channel. And in the middle of that, a really big protein, in the middle of that protein, there's a little channel where, oh no, I'm sorry, it's outside. There's lipid in the middle, but you've got like a 4-barreled channel, and each one of these barrels allows water in or out. So this is used in the kidney then when you want to regulate how dilute or concentrated your urine is. If your body is getting dehydrated, you pull these out of the membrane and your urine will get more concentrated. If you drink too much, like Lake Isaacson drinks too much, his body then releases or stops releasing antidiuretic hormone, and it puts these channels into the membrane of the collecting ducts, and water leaves and you get dilute urine. So, and that's why you pee a lot when you drink a lot of beer. You're bringing in a lot of fluid, so your body needs to get rid of that fluid. And also the alcohol stops the antidiuretic hormone from being released. So you get more dilute urine. And that's why when we were kids, we used to say, you're not drunk until you pee clear. You drink a lot of beer, eventually your pee is just clear. It's just water coming out. And that's because of this antidiuretic hormone effect. And that's why you're going to be hungover the next day. You're going to be dehydrated and a lot of other things in hangover, but that's a lot of it. Which, I should pause right now and just give you this sideline. When you've drunk too much, drink a lot of water before you go to bed. Also, some sugar, some salt in that water will help too. Salt and water before you go to bed. It's going to help that hangover the next day. The next day you're screwed. There's no way out of it. But if you do it the night before, you'll be better off. See, another little thing you get just for knowing me, just another little tip. Oh, you kids are so lucky. Okay, another thing are the porins. These are mainly prokaryotic, but they're pretty big barrel-shaped proteins that allow molecules to go through. So you see these a lot in like the outer membrane of the mitochondria and the chloroplasts, right? Because of that whole endosymbiotic idea, these are kind of like prokaryotes inside of our cells. So you see these porins in mitochondria and chloroplasts if you're a plant, which I'm not. Okay, so here's porins and aquaporins again. Coming through. What else? Okay, so CFTR, cystic fibrosis transmembrane regulator. So this is what can happen if you lose a transporter. There's one of those text boxes in your book that explains all about the CFTR. So basically, CFTR is a protein that looks like it should be an ATPase, a pump. It binds ATP, but it isn't a pump, it's a transporter. It allows chloride to leave the cell. And the basic idea is this is in the epithelial cells that are lining your bronchi and down into your lungs. And what this does is regulate the thickness of the mucus in your airway. So I think I talked about this when I was talking about cilia, but basically your epithelium there are constantly secreting this thin, watery mucus, and it lines that membrane. And anything you breathe in, dust, bacteria, viruses, there's a good chance it's going to get stuck in that watery mucus. And then you've got cilia here that are constantly beating in the up direction. So all that stuff is coming up out of your lungs, down the back of your throat and into your stomach to be digested and gotten rid of, right? So, and when you get a cold, one of your body's responses is to make this a little thicker, so it's more likely to grab onto stuff like that. And that's why when you have a cold, you're like, mucus coming out everywhere because it's trying to, stop the viruses from getting in. And, I just did it, bring some of that up from the back of your throat. I've got asthma, so I've got a lot of that going on a regular basis. So basically, that's how your lungs keep themselves clean, by releasing this thin, watery mucus. And the thickness of this mucus is based on sodium and chloride. So if we'll talk about osmosis in this chapter as well, but when solute goes, water follows it, and that's going to keep it thin and watery. So that's a normal person. Now, a person with cystic fibrosis lacks that transporter. Okay, so they can't let chloride go out into the mucus. Since chloride's not going out, sodium's not going to go either. Again, for that electrical reason. Once you get a bunch of sodium out here, the sodium here are going to repel more sodium from coming, and so you can't really keep pumping sodium out. Right? You can't put a whole lot of positive charges in one place. They repel each other. So in this situation, you're putting neutral particles out there because you have sodium and chloride. Here, neither can go because chloride has stopped. So what happens is your water doesn't go either. The mucus gets really thick, and bacteria get caught in it like they normally would, but instead of being brought out of the lungs, those cilia can't beat that mucus out. It's just too thick. And so bacteria gets stuck down here in the lungs, and you get really bad respiratory infections and typically die pretty young. I mean, if you don't have any treatment, I think the life expectancy of CF without treatment is probably about 10 or 12 or so. Maybe 20, I'm not sure, 20, 21 perhaps. But young anyway, these people die young. And again, it's just because of that one transporter that's missing. Now, when I was in high school, grade school through high school, I had a friend with cystic fibrosis. And he said every morning he'd lay over a chair, with his head down, laid over his chair, and his father would beat his back to get vibrations going, and all this mucus would come out, and he'd spit it out, spit it out, spit it out. And then, when he got home from school, they'd do it again. And it was a hard life. It was a really hard life. And he died, I think he was 21 or 22 when he died. So, I mean, today they've got more treatments for it, I believe. I'm hoping for a CRISPR solution to this. Get rid of the bad gene, put a good gene in, and fix it. But that's not what I do for a living. So I'm just hoping on it. Okay, so that is all of passive. That's all of passive transport. Okay, you've got simple diffusion and facilitated diffusion. Like I say, 45 minutes or a half an hour short, but that's typically what I cover at this point. So, are there any questions? Any comments?
What does the CFTR protein stand for again?
Cystic fibrosis transmembrane regulator. Cystic fibrosis transmembrane regulator. If you look at that box 8A, you'll see an actual drawing of it, see what it looks like. Mr. Mendoza. Darling Mendoza. It really stinks when your parents give you a name like Darling, because then everybody knows you. Someone like Robin Russell can hide. See, she's hiding now. You don't even know she's in the class. But Darling, I see the name. It's like, oh, yeah, I know that guy. Okay. Farouk, you're standing out right now, too. And DeJovany, oh, come on. You're going to stand out the rest of your life. I guess that's a good thing. It's a good thing.
Yeah.
I got to say, DeJovany, I absolutely love it.
It's DiGiovanni. You're saying it wrong.
DiGiovanni. DiGiovanni. Well, thanks for telling me.
Get him.
DiGiovanni. Okay. Absolutely love that name. All right. Any questions or comments here? Anybody else want me to make fun of their name? Barbara Perry, can I call you Babs from now on? Oh, I'm just killing time because I feel guilty that I ended so soon, but that's all we got for today, people. I hope to see you all again on Thursday. Have a great week.
You too.
Later, dudes.